So introducing the 2024 John Kendrew Award recipient, which is Thibaut Brunet. So Thibaut is evolutionary biologist. He has been interested in evolution from the very early stages in his career. Already at the level of a master's student, he has worked with Emmanuel Farge, where he really did fantastic work and published a very interesting, influential paper, which extended the observation of Emmanuel Farge that mesoderm specification is functioning, under control of mechanotransduction. He extended that to zebrafish, showing that this kind of mechanogenetic feedback is a conserved feature. Then Thibaut came to EMBL, to the group of Detlef Arendt, and he has done here a fantastic work on the evolution of bilaterian musculature and the notochord of axolotl, which I learned from Detlef, is called axochord. And I would say a little bit more about that later on. As this kind of, as has been always almost required for German scientists in America gewesen. So Thibaut is French, but he went to America and he went to the lab of Nicole King, and there he did some really transformational work about the evolution of morphogenesis and about the collective contractivity in choanoflagellates. And he continues doing this kind of work now at his own lab at the Institut of Pasteur. He keeps focusing on the choanoflagellates. I want to tell you a little bit more about his, really transformational work, which he has done in America. So in developmental biology, we think a lot about morphogenetic modules, about how gene regulation kind of governs effectors which are able to change cell behavior, and how those are collectively kind of coordinated to create changes in shape of tissues or organisms, right? One of these morphogenetic modules is shown here in the middle, kind of, put more or less in the context of Drosophila, and what it results in is a bending of the tissue. This is something which is happening in many different developmental contexts, and it was kind of thought to be really an invention of animals. So what Thibaut did was to show that the closest animal relatives, the choanoflagellates, which are typically unicellular, but they can also form multicellular colonies, they have similar system. They are also able to bend the colonies in various ways. But what was very interesting about his discovery was that the context has changed really dramatically, because everything is different. While in animals, such process is regulated by transcription factors and signaling pathways, in choanoflagellates, it is regulated directly by light. This is related to the function of this process. In animals, it is about shaping embryos typically. In choanoflagellate, it is about changing the behavior of this colony from being able to feed or being able to swim away from, let's say, a predator. What they might have in common is this mechanism, which is based on contractility of actomyosin, but the details of that are still not completely clear. And so basically everything changed, but the only thing which actually remained as a constant is the need to bend the tissue. And so that kind of indicates, that these kind of physical requirements, might be something which is actually driving, evolution of these systems. And that, in fact, this is not invention of animals, that the components is always puzzling to people, that the components for this are present in the eukaryotic unicellular sister groups, but they, are not used for something like that. But Thibaut showed that it is. So this was a big deal. I personally think that this paper is absolute tour de force, because Thibaut, he found that choanoflagellate, which makes the colonies in a pond in Curaçao. He caught it. It's a new species. He actually named it, right? But then within 2 years, he has done, so many, innovative experiments, which he will probably tell us about. For example, he manipulated gene expression or gene function in the system, which is a non-model system because it's a new species which he just caught, right? He manipulated it by manipulating the microbiome or what they eat, right? And like that, he kind of knocked out the pathways which are able to sense the light and change the behavior of these animals. So very fascinating stuff. Then they also expanded to other choanoflagellate species, really did a phylogenetic analysis, and they really drove this home. Why do I go into such detail about this paper? Because I actually wrote a little kind of insight on it for Science. This was the Science cover story. It was published. I mean, you can read it. It's called Evolutionary History of Tissue Bending. But continuing on the theme which we had just, essentially finished with Matthias. And now this is not a joke, right? When I wrote it, I gave it a very different title, and I show you how it was. Bended like a choanoflagellate. Right? I mean, you get it, right? I mean, who doesn't get it? Right? The Science editor did not get it. And I fought for it. I sent her the movie. Everything, but it was apparently not possible. So I'm really sorry about that. This would have been so much better title for this news and views. But anyway, this kind of brings me to say something about Thibaut. What I think. I'm very active on Twitter, and when something comes up which I'm very interested in, I call it a stuff that matters, right? And so Thibaut definitely works and does stuff that matters. I have spoken to Detlef about how he was here at EMBL, and Detlef said that he is a scholar, that he is extremely well read, that he's actually probably the most knowledgeable living zoologist of these days, right? And that comes from Detlef Arendt, right? So, okay, that's a really, really strong endorsement. I mean, of course, you can see that because Thibaut has written many very thoughtful reviews about the stuff that matters. And I know him mostly from the meetings, from the circuit. What I observed about him is that he actually is able to multitask amazingly. He listens to a talk, and at the same time he types with 2 fingers, right? But he doesn't look at the keyboard at all, right? And he essentially stenocopies what the people who are giving the talks are saying. Absolutely incredible. I want to also say a few fun things about him. So I learned that he likes to whistle. I think that might be a French phenotype, because I think one of the senior members of the audience here also likes to whistle, and he's also French. Do you know who this is? Yeah, Eric is smiling. Yeah. Eric Carcenti, of course, likes to whistle. When I was here a PhD student, you whistled. You still whistle. I saw you today whistling. Thibaut apparently can whistle, operates his vocal cords so fantastically that he can imitate a cell phone ringing, right? So in the Detlef Arendt lab, he was kind of creating chaos by imitating the ringtones of the phones of various people. So incredible. And he really has a great voice because he sings in the choir. So I will essentially stop here. I forgot to put this on, but I will do it actually now, right? So what I want to say at the end is that, yeah, I mean, Thibaut is really deserving person to receive the John Kendrew Award. He works on stuff that matters, and he reminds us what it is to be a scientist, researcher in biology, that we can really learn enormous, new, interesting, spectacular, impactful things by looking at new animals, by looking at the biodiversity, by going away from all these areas where we have to compete with thousands of other labs, by looking at something really unique and novel. And we can really, this is what makes us biologists, this is what we should focus on, to really learn from nature, what different ways it has evolved, to solve problems, right? And for me, Thibaut is an impersonation of a person who does this, and for that, I put my hat off for Thibaut. Now I will give you a medal. Maybe. So, start with the medal.
All right.
Okay. And we take a picture. You open it.
All right. What about you?
Don't be nervous. I'm also nervous. This didn't. Okay. So, Thibaut, now you have 20 minutes to tell us about your amazing science.
Thank you so much, Pavel. I will never live up to Pavel's extremely generous introduction, but I'm extremely touched, honored, grateful to receive this award. The EMBL is the place I was born as a scientist, so this really means a lot to me. We'll tell you a little bit about the research we do, trying to bridge concepts of cell biology, evolutionary biology with new organisms that we sample in the wild. But of course, I wanted to start with the most important slide and to emphasize that research in experimental biology is never done alone, and at least in my case, I've done everything always in collaboration, so I want to acknowledge my mentors over the years, including, of course, especially Detlef Arendt and his lab, but also Nicole King, and of course my current lab, and you can in Institut Pasteur, and you can see us here engaged in a weird ritual whose significance will become clear later. And so our key question, as Pavel mentioned, is how animal morphogenetic mechanisms evolve. So all animals look the way they do because they underwent morphogenesis during embryonic development. And the mechanisms of this morphogenesis rely on a small number of conserved modules that are present all across the animal tree of life, which include simple things like multicellularity, cell adhesion, collective contractility, some types of tissues like epithelia. And what we want to understand is where these come from. And since all of these features are shared between all extant animals, it means some version of these complex features was already present, most likely in their last common ancestor. An important thing to keep in mind is that animals have evolved multicellularity independently compared to, for example, plants and fungi. So that implies that if we want to understand where these features come from, how they evolved before the animal common ancestor, we have to consider information from neighboring branches of the tree of life, and notably from the organisms we study, which are the sister group of animals, so they're closest living relatives, which are called the choanoflagellates. And so choanoflagellates, if you've never seen one, choanoflagellate is, they're often unicellular. As their name indicates, they're flagellates. So they have a flagellum exactly like a sperm cell with which they swim. But they also have an additional feature, which is a ring of filaments called microvilli around the flagellum, which is called the collar. And the flagellum and the collar together form a collar complex, which is fundamental to the life of choanos in two ways. The first way is that the beating of the flagellum allows them to swim around, exactly like a sperm cell. But it also creates a flow of liquid that carries bacteria that are phagocytosed by the choanoflagellates. So this is how they feed, and we always culture them in the lab together with bacteria that are also an important source of signals, actually, for their biology. So there's not only one species of choanoflagellates, we know about 400 of those. Here is a very old depiction of the diversity of choanos, where you can see that there's a diversity of unicellular form, some diverse multicellular colonies as well. You can also observe that diversity today with modern tools, and you can see here a modern selection of choanos. Some of them are unicellular, some are multicellular, shaped, for example, like chains, some like rosettes, some a bit more like these tufts of tentacles here. So there is a diversity in morphology and multicellular development across choanos. And of course, we're always interested in developing new species as models. The best established one is this one that you can see here, which is called S. rosetta. So it develops into these spherical colonies that look a bit like blastula stage embryos. It's become genetically tractable a few years ago. But S. rosetta has limitations. Notably, it was only found once in the wild. It could never be isolated again. So its natural environment environment and the natural function of this multicellular development is actually a little bit unclear. And in part, to expand our knowledge of coenos and also to compensate for these shortcomings of diverse models, we've been isolating new species of coenos. And what I will tell you about in the rest of the talk is about this species that Pavel introduced called C. flexa, which we found back in Curaçao, so in the Caribbean, back in 2018, with those people, Nicole, Ben, and Tess. And this coenoflagellate actually makes colonies that are fairly different than the rosettes I presented you earlier. So in this case, it's a monolayer of cells, a bit like an epithelium, and all the cells are connected by direct adhesions between their microvilli, so between these collar of filaments that surround the flagellum of each cell. And this monolayer is actually curved, so it's like a cup if you want, and all the flagella are pointing toward the inside of the cup. But one very interesting and striking thing about that species is that that cup can actually change shape. In a matter of seconds, that cup is capable of inverting, a bit like an umbrella in the wind, if you want, so that the flagella that are pointing inside point out, and it can go back reversibly to the flagella in configuration. So if you wonder what this looks like in live imaging, here's a video. This was actually captured in the marine biology station in Curaçao. So you see here these colonies with flagella pointing in, they contract, the flagella point out again, and then this behavior is reversible. So people sometimes ask me how we felt when we first saw this behavior, which was a form of collective behavior that was not expected from coenoflagellates, that was not known before. In fact, I don't really remember how we felt. It's a bit of a blur. But the good thing is that I don't have to remember, because the camera we used in the marine biology station to record this movie was actually, and we had no idea that was the case, capturing sound. So I will show you, I thought this was an occasion to show you a short extract of the unedited video where you can see the behavior of the coenos and hear the behavior of the human beings around. And so you will hear Ben Larson speaking and me making some comments.
All right, Thibaut.
Yeah.
I got it.
Yeah, show me.
Well, I don't know how to watch these videos again, but I got one expanding now.
What nice.
And I also got the contraction. And see, look, cool. Now it's showing the morphogenesis. Yes!
Perfect.
Flip inside out. It flipped inside out.
Whoa.
Yes, there it goes. Yes! We got it.
Perfect.
Now it's inside out. Yes! Yes! And now hopefully it switches back again. Please switch back again. Oh, it's switching back. Thibaut, it's switching back. It is. It's switching back. It's stuck. It's gonna flip inside out. Here it goes. Yes! It did it. It just did it again. Yes! Yes! We got it.
You can safely anticipate many such cycles.
Yeah! Yes! Look at it. There it is. Back again.
One cycle.
One more cycle. Keep going, you tiny animal.
Anyway, I'll stop here. It continues like this for 20 minutes. At some point, one of our colleagues walks in and calls us huge nerds, probably for good reasons. And so anyway, we were very fortunate that we could bring this species back in the lab. We could culture it. We've now sequenced its transcriptome, sequenced its genome. We are starting to be able to manipulate it genetically. And so some of the first questions we addressed are, how do we control this behavior? And are there stimuli that allows us to induce this inversion? And we found that actually it responds to quite a lot of signals, including light, and more precisely light to dark transition. So you see here a population of colonies that are relaxed with their flagella in, and you will see the light will go off at some point, and almost all of them start contracting and expose their flagella towards the outside. And we could, to some extent, figure out the molecular pathway that detects light, which relies on a rhodopsin coupled to a phosphodiesterase, so this induces inversion. We could also show more recently that some chemical signals contribute, like nitric oxide, which is a very interesting molecule because it's a paracrine signal in the human body, and we think it likely acts also as a paracrine signal in C. flexa. And here again, we did some biochemical experiments to characterize the pathway. It also responds to mechanical stimuli. And you see here an innocent colony that doesn't know it's going to be poked, but here it is. And so they also respond very exquisitely to touch and to flow. And for this, we have no idea of the mechanism. There are actually too many candidate pathways. But one interesting question that we are now addressing in my lab is whether this provides coordination between cells. Do they sense their neighbor's deformation, or do they use that to consolidate this inversion behavior? And one other thing we did is that once we had experimental control over this inversion behavior, we did some experiments to figure out the mechanism, and that's something Pavel mentioned. We think that apical actomyosin contractility plays a part in this. We think it's not necessarily the full story. There are likely several mechanisms at play, and this is something we're characterizing more in depth still now. And finally, of course, one simple question which was important was, why do they do that? What is the function of this behavior? And in fact, we could show that we're pretty sure this implements a trade-off between feeding and swimming. So the form with flagella in, which is relaxed, feeds very well, but it doesn't swim around. The form with flagella out swims very well, but it stops feeding. So in a way, this inversion behavior can be interpreted as an escape response. When things are good, you stay where you are and you feed. When things are less good or you perceive a stressor, you invert and you swim away. So this was the original reason we decided to study C. flexa. It was to understand this very unusual collective behavior. There was one question that we never really addressed in the past, which was the question of development. How does it go from a single cell to a multicellular colony? And the reason we didn't address it was that we thought we knew the answer. And I will tell you a bit more about this later. But that question was tackled by two very brave students on postdoc, respectively, Hosan and Nuria. So these are really the heroes of this story. And in fact, if you take a step back, basically there are two ways for microorganisms to become multicellular that are classically recognized. One way is clonal development. You divide, and by serial cell division, you become a colony or an organism. This is the way animals develop. This is also the way all other choanoflagellates that had been studied in the past developed. So we expected, of course, it's going to be true as well for C. flexa. But in fact, there is another potential mechanism, which is aggregation. So if you have several cells that are solitary, that live independently, in some conditions, they can come together and become multicellular. And that's known, for example, in social amoebae, like Dictyostelium, you heard about from Jordi this morning. And so these two modes of development, of course, have very different constraints. Clonal development is constrained by the rate of cell division, so it's kind of slow. But it has a big advantage, which is that all the cells in a clonal colony are genetically identical. So there's very little genetic conflict. On the other hand, aggregation can be fast if the cells are dense enough. They just have to come together and to adhere and to self-organize. But you can combine different genotypes by aggregation, potentially different strains, and that creates genetic conflicts that raises the potentiality of exploitation by cheaters. So potentially because of that vulnerability to cheaters, it's thought that aggregative forms never become very complex, unlike animals and plants that develop clonally. So anyway, we were fairly convinced that C. flexa would develop clonally, but some observations were a bit perplexing, and that motivated us to actually test that assumption. So we isolated a single cell. That's much harder than it sounds, also hard to immobilize them. But anyway, we could show that if you isolate a single cell, in fact, they can perfectly well develop clonally. So if you image for 18 hours, if you manage to book the microscope for long enough, you can see a single cell develop into a small colony of about 6 cells that has a perfectly canonical morphology and behavior. But now if you perform another experiment, if you dissociate C. flexa into single cells, you find that actually those cells come back together in a matter of minutes, and they can reaggregate. And even if you mix two different populations of cells marked in green and pink here, respectively, they will come together and form colonies that, if you wait just one day, are clearly chimeric, so they are clearly formed by aggregation. These cannot have developed clonally, but they have a perfectly normal morphology and behavior of C. flexa colonies. So it was a bit strange for an organism to be able to engage in both of these behaviors in parallel. We think in reality they often develop by a mixture of aggregation and clonality. It was even stranger than a choanoflagellate, for a choanoflagellate to do that. So that motivated us to address the question of why, again, do they do this? And of course, natural biological phenomena are often better understood in their natural context, and EMBL has been a pioneer of planetary biology over the past few years. So that motivated us to go back to the natural environment where C. flexa had been found, which was this Caribbean island.
No, I don't die.
No, I don't. In fact, evaporate over time. So these are very ephemeral environments, and then very often they even completely dry up. So if you follow, we went to the field back last year, we followed about 100 of these splash pools, and we can follow that the depth decreases as they evaporate until the waves come back and they get refilled and the cycle restarts. And salinity follows the opposite trend. As it dries up, salinity goes up and until it's dry, and then the waves come back and salinity goes down again. And so because of that process, at any given time, there's a population of splash pools that are at different steps of this cycle. So if you plot now the salinity of splash pools, you have them here simply in order. So the least salty is a bit less salty than the seawater. The saltiest are at saturation, basically almost dry. you see that there's a range of salinities across these splash pools. And if you look now at where C. flexa is found, it's found in a very narrow range of salinity, which is basically between seawater and about 3x seawater salinity. So that made us wonder, how is this development of flexa constrained by these changes in salinity? And in fact, to do that, we built artificial splash pools, so just an experimental setup in which we matched the rate of evaporation to the rate we had measured in the field. And I'm going to skip a little bit ahead, but basically if you follow what happens to colony when evaporation, when salinity crosses a critical threshold beyond which we no longer find colonies in the wild, these colonies, I'm going to play this again, actually dissociate into single cells. So we would no longer recognize them as C. flexa colonies if we found them in the wild, notably because if you look now at these single cells, they've completely lost their flagellum and their microvilli that are normally what allows us to recognize choanoflagellate. So they become round. And of course, you might be wondering, are these cells simply dying, or is it something like a cyst that resists high salinity? And one way to figure out is to rehydrate those cysts. You can actually dry them up completely, so you can make them into powder. And if you rehydrate them and wait a few days, you actually find that these cysts have developed back into perfectly convincing colonies. So to conclude, I don't want to be too long, and we also did the same in the wild, with pools that had evaporated, and we scratched the soil or the salt. So this is Nuria here scratching the soil. And if you rehydrate them after a few days, you can recover perfectly convincing colonies from this salt. So basically, the model we have in mind now, it's a working model that we're still busy testing, is that this multicellular life cycle is untrained by salinity fluctuations in nature in such a way that dry splash pool contains cysts that hatch when they get rehydrated by the waves. The single cells then basically are engaged in a race against the clock to develop multicellularity while salinity is still permissive and compatible with feeding, compatible with division. So, and we think this is one way to interpret why they use both aggregation and clonality. They basically use every single possible mechanism because they have a very short time to develop to develop multicellularity. So they combine aggregation, development, and sheet fusion until salinity crosses the fatal threshold, and then they dissociate again, and they become cysts, and they wait for the next wave to come back. So we think this is an example of a non-environmentally entrained life cycle. So with this, I want to thank again everybody who contributed from EMBL, elsewhere, from my current lab as well. The preprint is on bioRxiv on the life cycle. I want to thank our current collaborators. We have many, notably in EMBL here, so I put them in green. So Gautam, Alba, Yannick Schwab, also Alexandre Pay, actually, still Detlef. And with this, I'll be happy to take any questions.
Questions for Thibaut? There comes Matt.
Beautiful story. So I was wondering about the dispersal of these. Do you think they spread out from these tidal pools, these splash pools? And have you tried sampling, perhaps through sequencing, in the surrounding ocean currents?
Yeah. So they do disperse in part by the waves, in part because the cysts probably get blown by the winds. There are also animals that go between splash pools, like crabs that can carry things. We've done some sequencing between splash pools. Even two neighboring splash pools can have fairly different genetic genotypes, actually. So there is some structuration to the population. In the open ocean, we think they're probably at very low abundance. We think they really thrive in the splash pools. So we could not find them in the Tara database, for example. So we think they're probably there, but just not very abundant. So yeah, what happens in the ocean, I don't know. Yeah.